Hello dear students. Today we are going to discuss the topic Brassinosteroids, their brief introduction, discovery, chemical nature, bioassay and their physiological role. The objectives of the present study are: first, to gain knowledge about the discovery of brassinosteroids; second, to know about the chemical nature of brassinosteroids; third, to study about the bioassays of brassinosteroids; fourth, to gain an insight into the physiological role of brassinosteroids and to know about the applications of brassinosteroids in agriculture and horticulture. Now, let's start with a brief introduction about brassinosteroids. Brassinosteroids are a group of plant hormones with significant growth promoting activity. They were isolated and characterized from the pollen of rape plant, that is Brassica napus. Subsequently, they have so far been reported from about 60 plants and are regarded probably ubiquitous in the plant kingdom. They are considered as hormones with pleiotrophic effects, as they influence varied developmental processes like growth, germination of seeds, rhizogenesis, flowering and senescence. Brassinosteroids also confer resistance to plants against various abiotic stresses. They are found at low levels in pollen, anthers, seeds, leaves, stems, roots, flowers, grain and young vegetative tissues throughout the plant kingdom. Brassinosteroids are a family of about 60 phytosteroids. Something about their discovery. Plant pollen has been considered for a long time to be rich in compounds with growth promoting activities. In 1970, an oily extract from pollen of Brassica napus was shown to cause extreme elongation of the internodes of pin to bean. A major screening effort was mounted by the US Department of Agriculture. Pollen extracts from some 60 different species were tested in various bioassays and shown to have varying amounts of growth promoting activities, with pollen from rape and elder showing some of the highest activities. The identities of the active compound in these extracts were unclear and were collectively called brassins, so called because the first extracts were from the genus Brassica. That they were not gibberellins, as indicated by some differences in growth pattern such as swelling and curvature of stem, which do not occur in GA-induced growth. A large-scale extraction and purification program was undertaken. Some 227 kg of B collected rape pollen was extracted and fractionated with activity tests at each step. Finally, 4 mg of crystals were obtained and subjected to X-ray crystallographic analysis to determine the structure. In 1979, this biologically active plant growth promoter was named as brassinolide and was found to be a steroidal lactone with an empirical formula of C28H48O6. Brassinolide was the first plant steroid with hormonal activities. In 1982, another steroidal substance with growth promoting nature was isolated from the insect galls of chestnut and named as castosterone. The discovery of brassinolide and castosterone gave an impetus to the idea of the presence of steroidal hormones with growth promoting nature in the plant kingdom. Since then, extensive studies on isolation and identification of brassinolide and its related compounds from various sources have resulted in the isolation of more than 40 analogues of brassinolide from more than 60 plant species and virtually all types of tissues including pollen, seeds, leaves, stems, roots and flowers. As the first steroidal hormone with growth promoting nature was obtained from Brassica napus, the name brassinosteroids was given to this new class of substances. However, brassinoline remains the most biologically active brassinosteroid and is widely distributed throughout the plant kingdom. Now, coming to the chemical nature, that is the basic structure of brassinosteroids. Brassinosteroids are a group of naturally occurring polyhydroxysteroid hormones with a chemical structure similar to the steroid hormones in animals. They contain the typical steroid nucleus with fused rings A, B, C and D and an alkyl side chain at carbon 17. Both the nucleus and the side chain contain various substituents in different isomeric configurations, making the stereochemistry and nomenclature highly complicated. Natural brassinosteroids so far identified have a common 5-alpha cholesterol skeleton, and their structural variations come from the kind and orientation of functionalities on the skeleton. The compounds can be classified as C27, C28, C29 brassinosteroids depending on the alkyl substitution pattern of the side chain. Brassinosteroids can be derived from the 5-alpha cholesterol carbon skeleton bearing the following structural characteristics. First, ring A mono to trioxygenated, always oxygenated at carbon 3rd. Second, ring B presenting a 6-oxo, 7-oxalactone or a 6-keto function or saturation. Third, all trans junctions of rings A to D. Fourth, 22-alpha, 23-alpha dihydroxylated, mostly alkylated at carbon 24, sometimes methylated at carbon 25 and sometimes unsaturated between carbons 24 and 28. These characteristics lead to considering natural brassinosteroids as the 3-oxygenated 5-alpha cholesterol 22-alpha, 23-alpha diols of plant origin, bearing additional alkyl or oxy substituents. They can also occur conjugated especially with sugars or fatty acids. Brassinosteroid analogues or compounds that show any structural similarity with natural brassinosteroids and or brassinolide activity. Natural brassinosteroids were isolated or detected in algae, pteridophyta, gymnosperms and angiosperms, indicating a probable ubiquitous distribution in the plant kingdom. Some of the biosynthetic precursors of the brassinosteroids such as cathasterone, 6-deoxocathasterone, 3-ip6-deoxocathasterone and 6-deoxo-28 norcathasterone as well as catabolites such as cryptolide or in some instances considered as brassinosteroids themselves, but they do not fulfill all the structural requirements. Some other natural products related to brassinosteroids have been reported, but their structures are incompletely elucidated. Due to the very small amount in which brassinosteroids are found in plants, special methods were developed for their detection and identification, as their isolation in pure state would demand a great amount of plant material and be very tedious and expensive. The biological detection of brassinosteroids was initially performed by the bean second internode assay, a test gradually substituted by the rice lamina inclination bioassay and the wheat leaf unrolling bioassay, while the immunological methods are less used. Due to the need to detect brassinosteroids in plant sources, a micromethod was very quickly developed for screening and quantification of these kinds of compounds that consists in reacting them with some chemicals to obtain derivatives. The derivatives are analyzed by gas chromatography with retention times sensitive to small variations in the structure of the brassinosteroids. They are also analyzed by mass spectrometry and selective scan ion monitoring. The latter method is being routinely used in the detection of brassinosteroids because its detection time is less than 10 pg. High performance liquid chromatography is the method of choice for final purification in the isolation of natural brassinosteroids, but it's unusual for their detection in plant sources. Reversed phase high performance liquid chromatography is less sensitive than gas chromatography for the detection and quantification of brassinosteroids. Now, their bioassays. Three common bioassays for brassinosteroids include the bean internode elongation assay, the rice leaf lamina inclination assay, and the wheat leaf unrolling assay. Now, the first, the bean second internode elongation assay. It uses bean, that is Phaseolus vulgaris, seedlings when the second internode from the root shoot junction is no more than 1 mm long, that is still growing. Gibberellic acids are active in this bioassay, but cause only elongation of the treated and upper internodes. Brassinosteroids, for example brassinolide, characteristically evoke elongation accompanied by curvature, swelling and splitting. Such a response is sometimes referred to as the Brassin response. Its magnitude depends on the brassinosteroid concentration. Thus, in the bean second internode bioassay, the brassinosteroid response is elongation, curvature, swelling and splitting of the treated bean internodes at hormonal levels of 0.01 to 50 mg. Although this test is sensitive to auxins, gibberellins and zeatins, only brassinosteroids are able to produce splitting of the internode treated. The reason for splitting is unknown, but probably is due to an imbalance among water uptake, turgor pressure and wall strength. Auxins are inactive in this bioassay. Now, the second, that is the rice lamina inclination test, a micro-quantitative bioassay for brassinosteroids. The rice leaf lamina inclination assay, which is used extensively for the identification of brassinosteroids during a purification protocol and determining the potency of natural and synthetic brassinosteroids, utilizes the bending of the lamina or blade of rice leaves away from the sheath when exposed to brassinosteroids. Explants consisting of lamina, lamina joint and leaf sheath from etiolated rice seedlings are floated on test solutions and bending of the leaf lamina is measured. In a modification, intact seedlings of dwarf rice Oryza sativa are used and test solutions are applied as a micro drop at the junction between the lamina and the sheath. Auxins may give a positive test, but at much higher concentrations than those of brassinosteroids. Significantly, however, application of auxin before, after or with brassinosteroid has a synergistic effect in that sensitivity to brassinosteroids alone. The reason for bending is not clear and may involve an epinestic response due to the production of ethylene. Gibberellic acids evoke a straight growth response without the bending characteristics of brassinosteroids. Now, coming to the third bioassay, that is wheat leaf unrolling test. It has been previously reported that brassinolide and its synthetic derivatives showed strong activity in the rice lamina inclination test and that this test can be used as a micro-quantitative bioassay of brassinosteroids. During further study of the growth-promoting activity of brassinosteroids on cereal plants, it has been found that brassinolide and its 6-keto congener castosterone were potent for stimulating the leaf unrolling of etiolated wheat seedlings, whereas indole-3 acetic acid, indole-3 acetonitrile and abscisic acid were inhibitory. A wheat leaf unrolling test is used to bioassay the biological activity of brassinosteroids and other phytohormones by examining the effect of brassinosteroids on wheat leaf unrolling in the following way. The brassinosteroids show strong activity in this test, a result completely opposite to the negative activity found for IAA. Seeds of wheat are placed on a wet cotton sheet for 24 hours. The germinated seeds are then planted in moist vermiculite and grown in darkness at 26 degrees centigrade for 6 days. Leaf segments are excised from the region 1.5 to 3 cm from the leaf tip under a dim green safe light. 10 of these segments are incubated in 1 ml of 2.5 mmol dipotassium maleate solution that contains a defined amount of the test sample for 24 hours at 30 degrees centigrade in darkness. The unrolling of the leaf segments was determined by measuring their widths with calipers. The widths of the control segments also showed increase from 1.2 to about 2 cm during incubation. Brassinolite and its 6-keto congener castosterone dramatically stimulated leaf unrolling, their activities being dose dependent even at 0.0005 microgram per ml. Both compounds markedly stimulated unrolling. At 0.01 microgram per ml and higher, brassinolite produced complete unrolling of the leaf segments to about 3.6 cm in width. Phytohormones like GA3 and cytokinins produced only slight unrolling. Zeatin had rather strong activity, leaf segments fully unrolling at 1 microgram per ml. Its lowest effective concentration being lower than 0.001 microgram per ml. ABA, IA and IAN tested in the concentration range of 0.1 to 100 microgram per ml showed negative activity in the bioassay. The slight unrolling of the control segments during incubation was avoided by treating with any one of these compounds. When used together with brassinolite, ABA and IAN reduced the promotive effect of brassinolite on leaf unrolling, but IAA showed no anti-brassinolite activity. When the wheat leaf unrolling test was compared with the rice lamina inclination test, the sensitivity of brassinosteroids in the former test was 1/10 of that in the later. The wheat leaf unrolling test, however, is a technically simple and time-saving bioassay because cutting wheat leaf segments is easier than cutting segments containing a second lamina joint as used in the rice lamina inclination test. The former test took only 24 hours, whereas the later needed 72 hours. Unlike the auxin-like activity of brassinosteroids shown previously in the rice lamina inclination test and in many auxin assays, brassinosteroids had activities opposite to those of auxins in the wheat leaf unrolling test. Thus, the wheat leaf unrolling test can be used as a simple and rapid bioassay for brassinosteroids and may prove useful for differentiating between brassinosteroids and natural auxins in crude plant extracts. The test is also a sensitive bioassay for zeatin because zeatin showed strong activity at concentrations of 0.001 microgram per ml or more. Now, coming to the physiological effects of brassinosteroids. Brassinosteroids have been found to have following physiological effects. First, stem and cell elongation. The most well-documented role of brassinosteroids is in promoting the elongation of stems, petioles and flower peduncles in dicots and of coleoptiles and mesocotyls in monocots. Exogenous application of brassinosteroids causes elongation in young growing regions in both intact seedlings and cut segments. Moreover, brassinosteroids are essential for stem elongation, as genetic mutants that are deficient in endogenous brassinosteroids have been found to be dwarfs than the wild type. Whether grown in light or dark, exogenous application of brassinolite or castosterone restored these mutant dwarf plants to the wild phenotype. Since the number of cells in the elongated organs or mutant was about the same as that in the wild type, The organ elongation by brassinosteroid application was not by increasing the rate of cell division but by enhancing the cell elongation. Brassinosteroids have been found to stimulate hypocotyl elongation in pak choi by increasing wall relaxation and a passive dilution of the osmotic potential of the cell sap. Cell elongation also depends on an adequate supply of new wall components and on the orientation of microfibrils and microtubules. Indeed, brassinolide alone or in combination with auxins enhance the percentage of transversely oriented microtubules. Brassinolide-induced cell elongation has also been suggested to be due to the reduced water potential of the vacuole via iron and or sugar uptake. Brassinosteroid and IAA effects are generally similar, but studies on carrot cell suspension cultures have shown them to act through different mechanisms. Brassinosteroid induces cell enlargement on auxin-starved cell cultures, but does not substitute auxin in stimulation of cell division. Now, second is inhibition of root growth and development. Brassinosteroids are powerful inhibitors of root growth, formation and development. Brassinosteroid and IAA effects are generally similar and a synergism between the two is typically reported. However, in case of root elongation and growth, they act quite differently. IAA is stimulating and BR inhibiting it. These differences may be probably due to brassinosteroid acting independently of IAA in roots or acting as an antagonist of IAA. As ethylene has an inhibitory effect on root growth and brassinosteroid stimulates ethylene production, it's possible that the inhibition of root growth is due to brassinosteroid-induced ethylene production. In general, exogenous application of brassinosteroids to root tissue inhibits extension and lateral root formation. Next is role in etiolation. Exogenous application of brassinosteroids show little effect on dark grown seedlings. However, brassinosteroid synthesis mutants, that's mutants which do not synthesize brassinosteroids, grown in the dark, show an unusual expression of features typical of seedlings grown in the light. For example, they lack an apical hook, have open expanded cotyledons from the first leaves and express genes related to photosynthesis and or anthocyanin production. This indicates that brassinosteroids have a positive role in etiolation, that is, they are needed for normal etiolated growth in the dark. In other words, brassinosteroids repress a precocious expression of photomorphogenesis in dark grown seedlings or in seeds germinating under soil. However, this phenomenon is seen in brassinosteroid deficient mutants of some plants, for example, tomato, Arabidopsis, but not in others, for example, pea. Next is effect of brassinosteroids on flowering. There has been very limited use of steroids in regulating flowering. The number of flowers in strawberry increased by the application of brassinosteroids at the foliage. However, in case of grapes, the application of brassinosteroids in autumn improved the number of flowers but inhibited if the time of application is delayed to late winter. Next is effect of brassinosteroids on senescence. Senescence is the process which refers to endogenously regulated deteriorative changes that become the natural cause of death of cells, tissues, organs or that of the whole organism. Like other hormones, brassinosteroids also play a crucial role in regulating the processes leading to senescence. The brassinolide promotes senescence in xanthium and rumex explants and in wheat leaves. In addition to it, brassinosteroids also accelerate senescence in leaves of mung bean seedlings. Besides, brassinosteroid deficient Arabidopsis mutants exhibit delayed senescence of chloroplast. Similarly, the senescence of the leaves of mung bean and mustard was delayed if supplied with 28 homobrassinolide at early stage of growth due to chlorophyll biosynthesis. During a search of senescence-associated genes, he et al. Developed a preliminary model for leaf senescence regulating network in Arabidopsis where signals such as abscisic acid, jasmonic acid, ethylene, darkness, dehydration and aging activated 147 senescence-associated enhancer trap line. 24-AP Brassinosteroid could activate some of these, but associated genes have not yet been cloned. Brassinosteroid also accelerates senescence in dying tissue cultured cells, and there is a delayed senescence in brassinosteroid mutants, supporting its role in senescence. Next is effect of brassinosteroids on photosynthesis. The aqueous solution of 28-homobrassinosteroid applied to the foliage of wheat and mustard or applied as seed-soaking treatment to mung bean, Lycoperdiscon esculentum, Raphanus sativus and dialkyl amino ethyl alkanoate ipibrassinosteroid in association with GA3 to spinach enhanced the photosynthetic rate. Application of 24-AP Brassinosteroid and 28-homobrassinosteroid through root was also reported to increase the net photosynthetic rate in Cucumis sativus and in Lycoperdiscon esculentum. Foliar spray of aqueous solution of brassinosteroid to wheat and mustard, 24-AP Brassinosteroid to seedlings of cucumber and brassinosteroid to rice increased the rate of carbon dioxide assimilation. Likewise, the foliar application of 24-AP Brassinosteroid enhanced the light saturated net carbon dioxide assimilation rate and carboxylation rate of rubisco, thereby increasing the capacity of carbon dioxide assimilation in the Calvin cycle. However, the epicotyle of cucumber did not respond to ipibrassinosteroid, but the transport of the labeled glucose towards the epicotyle was favored. Similarly, hill activity in the foliage of Vigna radiata was favorably affected on being supplemented with aqueous solution of 28-homobrassinosteroid. Next is effect of brassinosteroids on chlorophyll. The total chlorophyll content or its fractions increased in the leaves of Vigna radiata and Brassica juncea by 28-homobrassinosteroid and in Cucumis sativus by 24-AP Brassinosteroid applied directly to their foliage. Similarly, the values for the above parameters increased in the leaves of rice, Brassica juncea, Vigna radiata, Raphanus sativus and Lycoperdiscon esculentum raised from the seeds given pre-sowing treatment with 28-homobrassinosteroid. Moreover, the water-stressed wheat plants treated with 28-homobrassinosteroid possessed high chlorophyll level. Next is effect of brassinosteroids on carbonic anhydrase activity. Carbonic anhydrase is the second most abundant soluble protein other than rubisco. In C3 chloroplast, it is a ubiquitous enzyme among living organisms. It catalyzes the reversible interconversion of bicarbonates and carbon dioxide. The rate of conversion of bicarbonate to carbon dioxide is normally slow in alkaline conditions. However, carbonic anhydrase activates the use of bicarbonate in the production of carbon dioxide. In C3 plants, carbonic anhydrase has a close association with ribulose bisphosphate carboxylase where it elevates the level of carbon dioxide at its active site. An increase in the activity of carbonic anhydrase in the leaves was attained by the application of 28-homobrassinosteroid to the shoot of the Brassica juncea and through root dipping in tomato. Moreover, the seedlings of wheat, mung bean, radish and tomato raised from the grains treated with 28-homobrassinosteroid or 24-AP Brassinosteroid possessed high carbonic anhydrase activity in their leaves. Next is effect of brassinosteroids on nitrate reductase activity. The process of reduction of nitrate is catalyzed by the enzyme nitrate reductase, the level of which increased in the plants of rice, maize, wheat, Vigna radiata, Raphanus sativus, Lycoperiskon esculentum, and in the seeds of wheat by the application of brassinosteroids. Next is effect of brassinosteroids on the vascular tissue. The first report of a role for brassinosteroids in the differentiation of vascular tissues is that of Helianthus tuberosus. Cells transfer to the xylem differentiation medium in the presence of auxin and cytokinins will differentiate into xylem elements in 72 to 96 hours. Very few vascular elements develop in the first 24 hours following transfer into this medium. However, nanomolar concentrations of brassinolide included in the medium resulted in a 10-fold increase in xylem differentiation. This was observed in the first 24 hours. Also, significant increases in cell numbers were observed, indicating a role for brassinosteroids in cell division and differentiation. Homobrassinolide are also reported to increase the cell division in barley. Xenia elegans has been used extensively to study the formation of xylem or tracheidae elements, a process that has 3 distinct stages. Brassinosteroids have been implicated in the transition between stage 2nd and stage 3rd, where secondary wall formation and cell death occurs. It had previously been shown that the effects of uniconazole prevent differentiation of Xenia mesophyll cells into tracheidae elements, and this inhibition was overcome by exogenous brassinosteroid application. Uniconazole appears to suppress the transcription of genes involved in the final stage of differentiation, but could be recovered by the application of brassinolide. It suggests that brassinosteroids are synthesized immediately prior to secondary cell wall development and cell death and possibly induce entry into this stage. Next is effect of brassinosteroids on the yield of crops. Once the presence of brassinosteroids in plants was established, the next phase was to explore the possibilities of using these new substances in improving the yield of economically useful plants. Brassinolide has been used to improve the yield of lettuce, radish, and bush bean. Foliar application of dilute aqueous solution of brassinolide also improved the yield of wheat and mustard, rice, corn and tobacco. Brassinosteroids were also found to increase the growth and yield of sugar beet, legumes and rapeseed. Application of 28 homobrassinolide and 24 epibrassinolide significantly increased the yield of potato, mustard, rice and cotton, Vigna radiata, and that of corn, tobacco, watermelon, cucumber and grape respectively. Foliar application of brassinolide, 24 epibrassinolide and 28 homobrassinolide was highly effective in enhancing the yield of groundnut and tomato. Role of brassinosteroids in increasing the crop productivity of tomato is also reported recently. Moreover, in China, 28 homobrassinolide has been registered as a plant growth regulator in case of tobacco, sugarcane, rapeseed and tea flowering, senescence, chlorophyll and key regulator of cell differentiation. Next is effect of brassinosteroids on counteracting stress. Brassinosteroids have been reported to counteract both abiotic and biotic stress in plants. Brassinosteroids increased tolerance to high temperature in Brassica juncea. The ability of 28 homobrassinolide to confer resistance to stress in Brassica juncea has also been established. Application of 24 epibrassinolide have a protective role on shoot, root length, soluble protein, proline content and peroxidases in seedlings or brassicaceae under seasonal stress. Brassinosteroids have also been reported to have a variety of effects when applied to rice seeds. Seeds treated with brassinosteroids were shown to reduce the growth inhibitory effect of salt stress. When the developed plant's fresh weight was analyzed, the treated seeds outperformed plants grown on saline and non-saline medium. However, when the dry weight was analyzed, brassinosteroid-treated seeds only outperformed untreated plants that were grown on saline medium. Brassinosteroid-treated rice seeds considerably restored the pigment level in plants that were grown on saline medium when compared to non-treated plants under the same conditions. Brassinosteroids can also provide some protection to plants during chilling and drought stress. Next is promotion of cell division. Nanomolar concentrations of brassinolide resulted in significant increases in cell numbers in Helianthus tuberosus, indicating a role for brassinosteroids in cell division. Homobrassinolide are also reported to increase the cell division in barley. Restoration of leaf size after administration of brassinolide to the mutant DED2 and DWF1 of Arabidopsis could not be accounted for solely by expansion, supporting a role for brassinosteroids in cell division. 24-epibrassinolide can substitute for cytokinin in the culture of Arabidopsis, callus and suspension cells, and a particular cyclin gene, CYCD3, was upregulated. The mitotic rate increased in roots of wheat after treatment with 24-epibrassinolide, and the hexaploid triticum aestivum was the most sensitive. Volumes of nucleoli were also increased and the effects of cytokinin were similar. In synchronously dividing cultures of the algae Chlorella vulgaris, accelerated increases in cell number and marked increases in nucleic acid and protein levels followed brassinosteroid treatment. Significant interactions with auxin at very low concentrations of brassinolide were also observed. Besides above-mentioned role, brassinosteroids also play an important role in pollen elongation for pollen tube formation. Extract from the pollen Lichnus viscaria contains a relatively high amount of brassinosteroids. Lichnus viscaria increases the disease resistance of surrounding plants. 24-epibrassinolide, a brassinosteroid isolated from Aegle marmelos, was further evaluated for the antigen toxicity against malic hydrazide induced genotoxicity in Allium cepa chromosomal aberration assay. It was shown that the percentage of chromosomal aberrations induced by malic hydrazide declined significantly with 24-epibrassinolide treatment. Application of brassinosteroids to cucumbers was demonstrated to increase the metabolism and removal of pesticides, which could be beneficial for reducing the human ingestion of residual pesticides from non-organically grown vegetables. Now, lastly, coming to the future prospects of brassinosteroids. 35 years of research on brassinosteroids have brought into light several vital functions of this class of phytohormones in the regulation of plant growth, development and productivity. Further progress in the investigation of mechanism of brassinosteroids action in plants on the one hand and elaboration of economically feasible schemes of synthesis of natural brassinosteroids and their analogues on the other hand will surely make a basis for the inclusion of this new class of plant hormones in the regular package of chemicals used for optimizing agricultural production. Moreover, the knowledge of the physical and chemical properties of these steroids is tempting us to consider them highly promising, environment-friendly protectors and promoter of agricultural productivity. One of the major constraints to employ brassinosteroids at large scale in the fields is their high cost. However, recent progress in chemical synthesis of brassinosteroids and their analogues has led us to economically feasible approach that has brought practical application very near to the reach of the farmers. Pesticidal companies in China and Japan have started synthesizing brassinosteroids on a commercial scale. In India also, Godrej Agrovet Limited, Mumbai, introduced 28 homobrassinolide in the market. Brassinosteroids can act efficiently in plants as immune modulators when applied at the appropriate concentration and at the correct stage of plant development. Brassinosteroids are implicated in plant responses to abiotic environmental stresses and to undergo profound change in plants interacting with bacterial, fungal and viral pathogens. Brassinosteroids open up new approaches for plant resistance against unfriendly environmental conditions. With this, we come to the end of today's lecture. Thank you.